All right, guys. So one of the first lines of evidence that we're going to look at in terms of evolution is actually the direct observation of evolution in short time scales. So how is it even possible to see this within a human time frame? Well, remember that evolution at its most basic is change over time. So it depends on the organism that you're looking at, and it depends on the time scale that you're looking at. So let's take a few examples of well-documented direct observations of evolution, especially over short time scales, relative to millennia, I mean. So first we're going to look at Darwin finches, one of the most well-studied groups of birds in the world, pretty much. They exist only on the Galapagos, and they were key to a lot of Darwin's thoughts in terms of natural selection. So, looking at these birds, which have been well studied ever since the time of Darwin, any slight environmental change can have an effect on the overall population. So, if we look at a population of one species of Darwin finch, there exists natural variation within all populations. And the variation in this example is beak size. There are some individual birds that have larger beaks, like the one on the right, and some that have more narrow beaks, like the one on the left. So those with narrower beaks eat smaller fruit and smaller seeds, and the ones on the right can eat larger fruits and larger seeds. So there exists variation in beak size that's directly related to their diet. So what happened is about 30 years ago, there was a massive drought in the Galapagos. So as a result, a lot of plant species suffered and didn't grow effectively after the drought. And because those plant species serve as food for the birds, different birds survived differentially because their food sources had changed. So small-beaked finches, like the ones on the left, died out because the plants that produced the smaller fruits and seeds didn't grow after the drought. So all that were left were really big fruits and seeds that the larger beaked birds, like the one on the right, could take advantage of. So as a result, individual birds in a population with smaller beaks died. So they did not survive and they did not reproduce. But those with larger beaks did survive and did reproduce. So as a result, the population of birds for this species eventually, over a couple of generations, generally had larger beaks. And we'll talk about this type of natural selection a little bit later in a different part of the module. But over less than 10 years, you could see changes in beak size based solely on what's happening in the environment. Another example is a lot of islands, like New Zealand, for example, have what we call endemic species, species that you can only find on those islands. And as a result, a lot of those species do not have natural predators. So they're happy and healthy and living their lives without being threatened by predators. But also in places like New Zealand, they tend to get colonized a lot. So about 130 years ago, the British came and started colonizing parts of New Zealand and brought with them brook trout, like the one you can see on the bottom here. In a lot of the streams and rivers in New Zealand, these invertebrates here, with these little tail fins here, used to not have any predators. So they behaved differently. They were aggressive. They were more in open water. But once these predator fish were introduced, they would eat any and everything that they saw. So as a result, they ate the most aggressive and most visible stream insects. So the only insects that could survive in the streams were those that were shy and hid. So over 130 years, with the introduction of a non-native predator, a lot of stream insects changed their behavior. And behavior has a lot to do with survival and reproduction. If you can hide and you can be shy, you're all that's left. You're all that's left to survive, and you're all that's left to reproduce. So those are a couple of examples from what we can see in the field. What about in the lab? If you're going to look at evolution in the lab, you need what are called model organisms. You need organisms that are generally small-bodied and have a quick development time. Fruit flies, for example, can develop from eggs to adults within 3 days. Zebrafish, here on the bottom left, can develop in 2 days. And Arabidopsis, which is this plant on the right, can develop within a couple of weeks. And the reason that we use model organisms is because they have a small genome, they can develop quickly, and you can manipulate them experimentally to see changes in genomes, changes in behavior, changes in morphology over very short time scales in terms of weeks or months. Another ideal model organism are prokaryotes, bacteria and archaea, because prokaryotes are even simpler than the eukaryotes I just showed you. They have an extraordinarily simple genome, a circular genome, and it takes them minutes to go through asexual binary fission. They can divide quickly, rapidly, and you can get large populations, and you can manipulate the environmental conditions and look at evolution in the lab over the course of weeks with bacteria. That being said, in a lot of health care environments like hospitals, nursing homes, or whatever, we're actually seeing this evolution in action. We call it something different. We call it antibiotic resistance. Antibiotic resistance is the presence of a resistant strain of certain types of bacteria. Many of you may have heard of MRSA, which is methicillin resistance staph. And methicillin resistant staph, or MRSA, has a gene that makes it resistant to the antibiotic. There's other types of antibiotic resistance ones, but they all basically function the same. You have a resistant cell, and this little circle here is called a plasmid. A plasmid is a circular piece of DNA. And if that piece of DNA has a resistance gene on it, it can pass it to a different bacteria that does not. This is called conjugation. Conjugation is where plasmids with little bits of genes can go back and forth between different cells. So you could have a population of sensitive cells that get killed by antibiotics, and it only takes one resistant cell to pass on its plasmids to a whole population. So that's how MRSA and other antibiotic-resistant bacteria proliferate so quickly. This can also happen in eukaryotes. Pesticide resistance has been seen in aphids, which are plant pests, and you can also see it in a lot of weed species. All it takes is individuals that survive the pesticide or the antibiotics in the case of prokaryotes. All it takes is one to survive that has a resistance gene, and once it starts mating and reproducing, that gene proliferates, and you start to get populations of organisms that are resistant to the things that we apply in order to kill them. So this is becoming a problem in agriculture, a problem in healthcare, and everywhere else. And this is evolution over very short time scales. If we think about beyond resistance, if we think about some of the things that we take advantage of on a daily basis, the things we eat, the companions that we have, you can see short-term evolution in action everywhere. Take, for example, your friendly, happy, loving dog. When humans get involved in evolution, it's called artificial selection. Humans are notorious for taking plants, animals, and other organisms and domesticating them. Domestication is a form of evolution because it changes the populations of certain organisms. So all dogs are derived from an ancestral wolf species. They have a common ancestor to wolves. What has happened, though, is that humans have manipulated the morphology and the genome, because remember, the genome, even though you're not actually digging into organism cells and changing their genes, if you select for certain features, like if you think about the face of a pug or a bulldog, you're selecting for certain features, that becomes the only type of gene available that is then in these organism cells. Versus something else like a dingo or hounds or whatnot, these were all artificially selected for certain characteristics that humans wanted. Agriculturally, we do the same thing. If you think about some of your vegetables that you eat, broccoli, cauliflower, cabbage, Brussels sprouts, kale, or the very strange kohlrabi, all of these are the same species, but they have been artificially selected for certain characteristics. Broccoli has been selected for these really tiny buds that kind of erupt from the top. You can see that there. Cabbage, it's one giant terminal bud, meaning the top of the plant. And then all of these different things. So all of these terms here represent the part of the plant that was selected for by humans. And all of these are derived from the wild mustard plant, which looks nothing like them. But there are certain characteristics that make all of these plants have a common ancestor. That common ancestor is wild mustard, and we have selected for it. Wild mustard has a lot of sulfur compounds in it, so if you think about if you've ever cooked broccoli or cauliflower, cabbage, any of these, and let them sit, it smells awful, and that's because of the common history of sulfur-containing compounds in the wild mustard. And one of our most classic examples of domestication of wild species is the corn that we eat. This is teosinte, which is a Mexican grass. If you look at the seeds of teosinte here on the left in this red box, they're very small and very hard. But over time, humans domesticated the corn to ultimately have starchy, plump seeds versus these really hard, husky ones down here. And so this took thousands of years of domestication, but it was constantly selecting the plants that had the features that were wanted, and then breeding them back, and only breeding those plants that had the useful features to them. So through a variety of medical applications, human interference, and random mutation, we can actually see evolution in action.